Most visitors to Lynn Headwaters Regional Park walk right past this tiny creek. But to regional park staff, it's not just a creek. It's a treasure trove of biological data called eDNA.
eDNA is short for environmental DNA. So it's the DNA that comes off the skin cells and other bodily fluids of animals that hang out in aquatic ecosystems. And we're testing those ecosystems. So this is a new technique that we're testing out.
Water samples are gathered from creeks for lab analysis. Robin and her colleagues are looking for evidence of amphibian species that are considered at risk: the coastal tailed frog and the red-legged frog. The team must follow strict scientific protocol for gathering the water samples. This way of doing species detection has many advantages.
It's way less invasive than traditional methods such as catching and trapping animals, which is particularly important with species at risk because we want to cause the least impact to them possible. It's also less labor intensive. We just need some water. We don't have to set traps and come back tomorrow, and so it's a lot easier to do as well. A lot of parks, we don't know where all the species at risk are, and we don't know their important areas. So the more information we collect, really the better we can manage our parks.
This creek happens to contain visual evidence of one of the species the regional park staff is testing for.
Cool. There's a tailed frog tadpole.
Oh, amazing.
Yeah, he's just under that rock. See, he has a little suck face, so you can suck onto the rocks and hold on in the stream. So we should be able to find DNA if this tadpole's in the stream. It's a good chance that we're actually going to find something.
Keeping the water samples cold helps prevent the eDNA from degrading. Even so, filtering the samples has to be done as soon as possible.
We're filtering them through some flasks using a pump, and it draws the water through a filter with cellulose bottom on it, and that's what is actually going to catch the DNA. And that filter is what holds the DNA, and then we send that to the lab.
At the University of Victoria, a biochemistry lab does the highly specialized detective work of analyzing the water samples.
eDNA tests are capable of detecting down to one molecule within a sample. It's kind of like CSI for wildlife. With appropriate testing methodologies and appropriate quality control in both the field and in the laboratory, it's an amazing technique, truly transformative.
The lab report confirmed the presence of both red-legged and tailed frogs in Lynn Headwaters Regional Park. So staff will now use that information to help protect the amphibians.
We can also help educate the public about why to keep their dog out of the stream and why it's important to have the fencing. So it helps with education as well as our management. We're really happy that this project worked out, so we can try this out at other parks and other areas where we want to know what's going on, and this is a quicker, easier, and less invasive method to find out what species are present.